Hello and welcome to CRISPR Cas9, The Ride. We're so excited for you all to join us on this amazing adventure today.
So it looks like our boat has arrived. Please watch your step as you enter the boat. Keep your hands and feet inside at all times, and please remain seated. Now wave goodbye to the people on shore. Goodbye.
Bye. I'm one of your tour guides today, Delaney.
I'm Haley.
And I'm Henry.
All right, moving on. Oh, look, there's one of our researchers working on our DNA model. Hi, Joe.
Thanks to Joe, we can create a better quality of life by using the CRISPR system to our advantage. CRISPR is efficient, accurate, cheap, it can improve agriculture and research, and has been shown to treat viral infections, hereditary diseases, and even cancer.
Wow.
Overall, CRISPR has the potential to create many improvements in health and science and save many, many lives. Thanks, Joe.
All right, so you're probably wondering, what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats.
Try saying that 5 times fast.
You're telling me. And the Cas9 protein is the CRISPR-associated protein that enables the editing of genomes. So the way this process works is researchers develop a guide RNA sequence that binds to the target DNA sequence and the Cas9 protein. This RNA sequence then recognizes the target DNA, and the Cas9 protein cuts and deactivates it at the designated location. Other DNA sequences are then inserted or deleted in place of the cut DNA. So, a little more on CRISPR as we move right along, and its life-changing discovery. This technique was developed to combat incoming viral infections by taking snippets of the virus's DNA and creating CRISPR arrays of them. So CRISPR arrays are like antigens against an infection, and thus remember and attack the virus DNA if it invades the host again, in which Cas9 cuts and deactivates the virus DNA.
Good to know.
Yeah.
Now, if you take a look at our leafy timeline, don't worry, I'm not going to go through every single point, but I will say that from 1993 to 2005, Francisco Mojica and colleagues made continuous progress towards the discovery of CRISPR's function and its applications. In fact, as recently as 2013, CRISPR was actually used for human genome editing.
Wow. And as you look at this picture, you can see that we first started working on mice, moved on to zebrafish, then crocs and monkeys, and we're just starting to dip our toes into the human genome.
If you draw your attention off the starboard bow, you'll see the shipwrecks of some of the other gene editing techniques, talons and ZFNs. So how does CRISPR compare? CRISPR is easier to use, more efficient, and it's cheap. It has an accuracy rate of about 70%, while talons and ZFNs are only 1 to 50% accurate. 1%? Why even bother at that point?
Ridiculous.
As for cost, talons and ZFNs cost $3,000 to $7,000, while CRISPR plasmids only cost about $500 a piece. The price of a textbook. Am I right, folks? All you students know about that.
So true, so true. All right, and as you look to your left, you can see our beautiful agricultural fields created by CRISPR.
Hey Delaney, did you know, if you edit the lettuce genome, you can make it CRISPR?
Good one, Haley. Good one. Moving on with agriculture, CRISPR removed the gene responsible for dwarf albino individuals in rice. As you can see, the green long ones are much more useful.
And delicious.
Yes. CRISPR also removed the gene in wheat, making it resistant to powdery mildew. That's a good thing, not a bad thing. To powdery mildew, and it made a mushroom resistant to browning. This decreases the need for harmful pesticides. The mushroom is also the first CRISPR-edited organism to be passed by the USDA.
Wow.
CRISPR also increased corn yield during drought and increased crop production in tomatoes by increasing the genetic variation in them. Additionally, CRISPR increased resistance of M. bovis in cattle, which causes bovine tuberculosis. This experiment was important because CRISPR did not disrupt neighboring genes. Overall, CRISPR creates crops that last longer, survive in drought, and grow more. Millions of people are starving on this planet, and CRISPR has the chance to decrease that number. Just look at our smiling children enjoying eating our CRISPR-made food.
Oh, adorable.
Now, as we come around this next bend here, oh my goodness, are those pigs?
Aw, how adorable. Sadly, these pigs are being used for organ donation, but this helps save a lot of human lives. Pigs are used for farming human organs because they're similar to people's size and anatomy without being too close. So pigs have viruses in their DNA that could cause cancer in humans when an organ is transplanted. Luckily, the Boston biotech firm successfully used CRISPR to kill the virus in pig cells. The team now plans on making pigs' immune systems more similar to humans, so when an organ is transplanted, the human immune system won't attack that organ. So that's a huge thing. As you all know, there's a huge list of people waiting to get organs, and CRISPR can help save the waiting people.
As we continue our tour, we're going to pause briefly to discuss how CRISPR has the potential to cure infectious disease or viruses such as HIV. CRISPR successfully disrupted the HIV-1 provirus in human cells.
Human cells?
Human cells.
Human cells.
So the HIV-1 provirus inserts itself into the human genome and infects and causes harm to an individual. This research by Avina and her team inserted human cells infected with HIV into mice and were able to restrict the active provirus and block expression. They were also able to use CRISPR to create double-strand breaks at HIV-1 target sites, which self-repair through non-homologous end joining. This results in a frameshift mutation and removes the provirus from infected cells. This has the potential for the future elimination of HIV-1 provirus from infected individuals, which in the future can save lives. Avina and her team were also able to use CRISPR to target the LTR promoter that regulates the provirus and excise it from the genome, which prevented the reactivation of the provirus in T cells.
That is incredible.
Wow.
It is, isn't it?
So,
Now-
Haley, do you think that CRISPR can also be used to treat hereditary genetic disorders like Duchenne muscular dystrophy, DMD?
I believe it can.
You are absolutely right.
Wow.
Currently, DMD has no existing treatment. It can cause heart and respiratory failure in the mid-30s. However, CRISPR can repair the mutation in the dystrophin gene, hence the name of the disease, and restore dystrophin protein expression. Effectively, we can cure DMD and save lives.
Wow. Wow. Now, as we go around this corner, you can see Old People Island. These are some of our 180-year-olds who CRISPR has extended their lives on Earth. Oh no! Oh my goodness! Sorry, folks. Let's just keep on moving.
So Adrian, when DNA replicates, part of the telomere strands at the ends of our chromosomes shorten slightly. This can eventually lead to genetic complications. Our researcher Myrtle is in the hourglass there.
Hi, Myrtle.
How's it going? And she's showing how CRISPR can add telomere strands to the ends of our chromosomes, giving us the time we need. Who wouldn't want a little more time with their friends and family?
Wow.
The clock is
Good
ticking.
point. Good point. Oh my goodness! Shoot at children's eyes! It's one of our mutant robots attacking our researcher!
This is what our mutant mice actually look like, and they are completely harmless. Relax, everybody.
So cute.
They are adorable.
Aw.
So you're probably wondering, why do we even need these mutant mice? Well, if you direct your attention to that waterfall back there, you'll see the entrance to our secret lab. Now, in our secret lab, recently what we've done is add stem cells to mouse embryos. We then grow those embryos up and through multiple generations, we can knock out certain genes, but it takes over a year. It's a huge pain in the butt. So with CRISPR, we can just inject fertilized eggs with Cas9 messenger RNA and 2 guide RNAs, knock out multiple genes in one step, and we can see results in just a few weeks.
Wow.
And that accelerates research immensely.
Amazing.
Yeah. And using this accelerated research, we can now apply it to the future and helping humans. So CRISPR can be used to alter CAR T cells, which have been shown to be very effective in treating leukemia. It also treats Huntington's disease, perhaps. Huntington's disease is an incurable hereditary brain disorder that affects 1 in every 10,000 people.
That's important.
So that's really sad, but CRISPR has the ability to replace incorrect nucleotides on the HTT gene. This has been successfully tested in mice. So CRISPR is also being tested to modify the genes of mosquitoes to stop the malaria plasmodium from reproducing within mosquitoes.
Wow.
No plasmodium reproduction equals no malaria in humans. That simple.
Wow.
Additionally, an institute in Germany is working on using CRISPR to treat genetic blood disorders such as beta thalassemia and sickle cell disease.
Incredible.
Really amazing, saving lives.
So what about ethics, Delaney?
Now, overall, CRISPR has the ability to save human lives. Are you folks going to let your fears stop that? Well, we are here to ease those fears, so don't you worry. A lot of people are worried that CRISPR will be used for cosmetic purposes. And yes, CRISPR has the ability to create designer babies. However, regulation can be created to make sure CRISPR is only used for medical and research purposes.
Some other people like to bring up the idea of informed consent of future generations. Frankly, pardon my French, this is Ridiculous.
Henry!
I'm sorry, it had to be said. Everything we do, whether good or bad, is without the consent of future generations. So why does it matter if I edit my genome versus choosing a partner that's more evolutionarily advantageous?
You make a good point, Henry. Another concern for some is off-target gene editing, and as we discussed earlier, CRISPR is only about 70% accurate in site-specific editing, which result in about 30% off-target edits. However, TALENs and ZFN were between 1 to 50% accurate, so compared to these techniques, we have improved tremendously. Also, this is a fairly new mechanism for gene editing, so a lot of future research and advancements still need to occur to improve this process and make it more accurate and efficient. In the future, though, this process will result in saving many, many lives.
Yes. People are also worried that only the rich will have access to CRISPR, but as we've stated before, CRISPR is much less expensive than other gene editing techniques like ZFNs and TALENs, so it's actually surprisingly cheap.
So just to recap, CRISPR is efficient, accurate, economical. We can feed the world, get people off of the organ transplant list, eradicate disease, and extend our lifespans. In addition, it can drastically improve research in many, many fields. It can even bring back dinosaurs.
No, Henry, no. The genes are the DNA is a little too old in dinosaurs, but we can bring back mammoths, maybe some pigeons that we killed along the way as humans, other important crops, and other things. So yeah.
All right, we are coming to the end of our tour, and we would just like to say thank you to our sponsors. Thank you,
Thank you,
You're very kind. Thank you. All right, and it looks like we have reached the docking area. Thank you so much for joining us on this amazing adventure today, and we hope you all had a fantastic ride. Please watch your step as you step out of the boat.
Sorry. It's really important.
On this coated locust,
You work so well inside a scrapped out locust.